Hello everyone, my name is Joseph Bellissimo and along with Tianyu Liu and Reynald de Leon, I'm extremely proud to welcome you all to cGEM Toronto's 2020 PET project, Enzyme Engineering for Pet-Based Textile Degradation. The COVID-19 pandemic and global shutdown was particularly effective in emphasizing the threat and widespread nature of plastic pollution. Plastic production is increasing and current recycling solutions cannot keep pace with less than 10% of plastics being recycled. To make the issue worse, plastic production and the rate of plastic production is increasing with a 100 million ton increase in textile plastic production within the last 10 years. Even the gold standard for recycled plastics, polyethylene terephthalate, sees only 1 out of every 5 tons produced being recycled. Current popular recycling methods, such as mechanical recycling, result in lower quality with repeated cycles and can't deal with contamination, while chemical recycling is expensive and energy intensive. Thus, the cost of virgin PET is much lower than that of recycled PET, which further drives demand away from recycling.
So the question is, why enzymatic recycling? Well, a great quality of it is that it doesn't experience a degradation in quality with repeated uses. It can actually increase the quality as it turns otherwise low-grade PET into its constituent parts, ethylene glycol and terephthalic acid, which can then be polymerized into PET again, or used for something else entirely. It's more efficient in its ability to degrade PET when compared to chemical recycling, and it can deal with a greater amount of contamination as well as mixed plastics. We initially thought that enzymatic recycling would be best used for PET bottles, but as we spoke with more people working in the industry, we learned that mechanical recycling seemed to be sufficient, at least for the majority of recycling that's happening right now. This was discouraging at first, but as we continued to do our stakeholder research, we learned about other sources of PET that had no solutions. One in particular that drew our attention was textile waste. The plastic waste issue consists of many smaller units, one of them being textile waste. Our project hopes to address this, in particular the recycling of PET, otherwise known as polyester. Polyester production has been increasing the past few decades, and as of 2018 equaled 55 million metric tons, more than half of global fiber production. The overwhelming majority of the polyester that is produced will either end up in landfills or incineration. This is because conventional methods of recycling PET do not work for polyester. Now, this increasing production rate, along with non-existent recycling methods, mean that as Canada reduces the amount of waste we export, we also need a solution for this extra waste produced. We continued on exploring in the textiles industry, and we learned a few things that would later alter how we imagine all this to theoretically be implemented. It was brought to light that there are difficulties involving the collection and sorting of textiles. Coupling this with Christina Sadal's point about how donation centers are usually where they all go, we thought it'd be best to collect our feedstock here. Because our project is still in its infancy, we took the advice of Paul King to begin with a particular clothing item, in this case jerseys, as they're 100% polyester, and work out how the process would theoretically work within this narrow purview. Unfortunately, enzymatic recycling does come with its own shortcomings. We've determined that our enzyme would need to operate 36 times faster than the Tournier enzyme that is the fastest PETase produced to date, in order for us to break even.
Nonetheless, a biological solution can maintain the properties of the recycled plastic and is resistant to contamination and impurities, making it a sustainable long-term answer to the plastic problem. The biological depolymerization of PET is facilitated by hydrolase enzymes, particularly PETase and METase, which were discovered in 2016 and shown to convert PET to its monomers, ethylene glycol and terephthalic acid. Here we can see the structures of both METase and PETase. METase is composed of a lid domain, which is designed to specifically recognize the MET substrate, and a hydrolase domain, which catalyzes it into monomers. PETase is composed of only a hydrolase domain with a very wide active site cleft, which facilitates the entry and subsequent hydrolysis of up to 4 subunits of the very large PET polymer. The mechanism by which PETase, METase, and other PET hydrolyzing enzymes work is essentially conserved. A catalytic triad consisting of a serine, aspartate, and histidine residue utilize a protein transfer mechanism to hydrolyze ester bonds connecting PET subunits. Recently, a novel protein engineering attempt was made on the enzyme LCC, generating a variant with the highest PET depolymerization rate to date. Unfortunately, this enzyme is still significantly too slow at depolymerizing PET to be industrially competitive. Thus, we employed a novel protein rational design approach on the PETase, METase, and LCC enzymes in order to enhance their catalytic activity and thermostability to that required for cost-effective industrial integration. Our novel rational design pipeline facilitates the production of multi-mutant enzyme variants with diverse analysis across the entirety of the protein without having to rely on flawed energy functions. The result of this approach and further analysis generated the following top-scoring variants. Our Toronto PETase variant has 43 mutations from IS-PETase and a Rosetta energy score that is 20 kilojoules per mole lower, while still maintaining a very similar backbone structure. Here we show the docked PET4 ligand in the right figure, as well as all of the accompanying residues in its first contact shell. However, it is very hard to analyze this image, so we use the plot on the left, which shows the residues in both the first and second contact shell, as well as the number of interactions visualized by circle size. Now, there are many mutations, so our focus will be to point out mutations of great interest, or those that represent a common type or class of mutation that is seen often throughout the entirety of our protein variants. Here we show a critical mutation proximal to the active site, specifically serine 160, where we mutate an asparagine to a leucine at residue 182. The graph on the right shows that this residue is in a very nonpolar, low solvated area, so conversion to the nonpolar leucine facilitates enhanced hydrophobic effects. Here we show an opposite mutation where we go from the nonpolar isoleucine residue to the polar threonine residue. As we can see from the figures, this residue is in a solvated area and close to polar amino acids, so this mutation stabilizes the protein by enhancing hydrogen bonding. We also introduced an additional disulfide bond to our PETase variant, which is intended to stabilize adjacent beta sheets. Our Toronto LCC variant comprises 32 mutations from the initial Tournier LCC variant, which is the best PET depolymerizing enzyme to date. Our variant also has an energy 50 kilojoules per mole lower than that of the Tournier variant. In contrast to PETase, the docking of the PET ligand in the LCC enzyme takes on an almost 180-degree conformation rather than the more highly strained 90-degree conformation seen in PETase. This is due to a much more constrained and less flexible active site compared to that of PETase. Our analysis shows that a change from histidine to tryptophan to the key active site residue 164 enhances pi interactions with the second aromatic of the ligand, and since tryptophan is a much wobblier residue, it increases the flexibility and size of the active site, allowing the ligand to adopt a conformation that enhances hydrolysis. Here we see a mutation from threonine to cysteine at residue 188, which increases the proximity of the polar sulfur atom to nearby polar residues, and due to an active pH of 9, also enhances the formation of electrostatic interactions. Similarly to PETase, we also engineered a disulfide bond in our LCC variants to stabilize amorphous regions close to the N-terminus. Our Toronto METase variant contains 57 mutations from the wild-type METase and has a whopping 200 kilojoule per mole lower energy. Here we show the METase active site, which is primarily centered around the serine 225 atom in the hydrolase domain, but also has multiple interactions for residues in the lid domain. Here we show a mutation from threonine to alanine at residue 426, which improves the hydrophobic interactions between relatively all non-polar neighboring amino acids and lower steric strain as well. Here we show a mutation from an alanine to a proline at residue 493. Proline's fixed conformation is highly ideal at turns in the secondary structure. While our novel rational design approach did generate very interesting variants, its tools are limited to relying heavily on ancestral and existing sequences. Thus, we need tools like machine learning to explore areas of the sequence space that are very different from our existing enzymes.
To get a sense of how large protein sequence space is, we can consider how many possible sequences there can be for a protein of length 263, which is the length of our enzyme. So we have 20 possible amino acids per position, 263 positions, giving a total of 20 to the power of 263 sequences. That number is astronomical. Comparing this to all these other fairly large numbers, you can see it's not even a comparison. If you were to imagine the scenario where every single atom in the universe was doing computation at the speed of the fastest computer in the world, proposing 10 to the 16 sequences every second,
all these atoms have been running in parallel since the beginning of time, you would only be able to explore a tiny sliver of this space. Clearly, we need more efficient and smart algorithms to explore this space. One way we can explore this space is using machine learning. First, we train a generative model that can generate new protein sequences, shown here. Two, we train an oracle that predicts a function for any protein sequence. Then we generate sequences and feed them through the oracle to get its predicted function. Using this function, we rank our generated sequences, keeping the ones that score well and discarding the ones that don't score well. We then iterate. We retrain the generative model with this modified training set. The goal is, after rounds of iteration, hopefully the model will be able to generate sequences that better maximize our desired function. Why we decide to pursue this pipeline is actually motivated from the work we did last year, where we generated a sequence from the pipeline I just described. Even though the sequence has a very low sequence identity with the wild-type PETase, it was able to actually show some experimental activity. Preliminary wet lab data showed that not only was that generated sequence able to fold, it also had a better catalytic activity than the wild type. So this is promising. Can we make the model better? Just to recap, this is our generative model. It defines a probability distribution over protein sequences over which we can sample new sequences, as shown here. Here is the performance of our oracle that predicts catalytic activity for any given protein sequence. Here, what's important is to look at the performance on the orange dots, where that is the data that the model has not seen. It is critical that the model performs well, because this is how well it's going to discriminate between the generated sequences, the good and the bad. Thermal stability prediction does a bit better. However, one major limitation of this approach is that we're only giving the model sequence information. So how are we even sure that the sequences fold? And was that sequence last year just a fluke? So we can actually use information from protein structure here to guide and to act as a filter of the model's generated sequences. One algorithm we can use is called protein solver, which takes in a protein backbone, the geometry of it, and outputs sequences that are predicted to fold into that geometry. Using the sequences generated by protein solver, we can define a probability distribution over the sequence space of PETase. Then we can filter the generated sequences from our model based on the likelihood of the generated sequences under this distribution. Intuitively, the higher the likelihood, the more likely the sequence is to fold into the PETase backbone. One result from this improved model is shown here. Obviously, we will need further experimental validation to confirm the validity of the sequence. For more details of how our model works and additional modifications we made, please check out our wiki. Talking about structure, we can even validate catalytic activity using molecular dynamics simulations. Here is an MD simulation with the wild-type protein and its interaction with the PET ligand. On the right, you see a plot of the distance between the beta carbon of serine 160 and a carbonyl carbon on the PET ligand. The closer those two atoms are to each other, the higher the tendency it is for the reaction to occur. And so this distance can actually be used as a proxy for the catalytic activity of our enzyme. We did MD simulations on the design that's predicted to be the best design from the wet lab pipeline. And here we see the interatomic distance between the two key atoms of the design in red being much more stable than the interatomic distance of the same atoms in the wild type. So from the simulation, we can predict that the design indeed works and that the design has a higher catalytic activity and even stability than the wild type.
With our newly constructed protein variants complete, we have designed both an iterative and integrative hypothetical laboratory procedure. The iterative procedure will make use of in vitro p-nitrophenolbutyrate assays to empirically determine our enzyme variant's catalytic activity and differential scanning calorimetry to determine its melting temperature. Our integrative Our integrative pipeline makes use of lambda red recombinering, which allows for the efficient and stable integration of our enzyme variants into neutral loci, such as LAC-Z, in the MG1655 genome for heterologous expression. Our design consists of knock-in, knockout vectors with homology to our specified loci, and integration cassettes consisting of our protein variant with a LAM-B signal peptide, a T7 promoter and terminator, and parts of the LAC operon that facilitate IPTG activation. Now that we have assembled a bacteria that is capable of secreting these plastic-degrading proteins, we can throw them in a bioreactor. Here we show the design of the bioreactor system. Pressure, temperature, and pH are the three conditions of the reaction, which are indicated as Pi, Ti, and Ae in the diagram respectively. The product mixture, including our target product Eg and TPA, unreacted PET, E. coli cells, enzymes, LB nutrients, buffers, and other byproducts should be fed to the downstream purification process to isolate and purify the Eg and TPA. Here is the diagram of the purification process. The product mixture is first fed into a filter to separate the solid PET and TPA from the solution. Then the solid PET and TPA is mixed with phenol to dissolve PET, followed by another filtration to isolate the undissolved TPA. The solution above is then fed to a centrifuge to separate the suspended E. coli and protein from the liquid solution, which mainly contains Eg, LB, and buffer. Next, this solution is treated with LLE, using furfural as the solvent. The upper layer should be the aqueous solution, as the density of Eg and furfural is higher than that of water. After the LLE, a distillation column can be performed to separate Eg from furfural based on their different boiling points. Here we show the results of our cultivation simulation using Optimus Firm, which predicts complete substrate degradation in 10 hours in the top right graph and shows expected levels of TPA and Eg in the lower graphs respectively. Here we show our final product amounts and properties after purification using simulations generated via Aspen+. The composition of each stream in the purification process is summarized.
So how would all this work in practice? Well, this diagram explains it. We would begin with our feedstock that would be collected from donation centers. Then they would be sorted, decontaminated, and granulated into PET pellets that could be efficiently used by the bioreactor. Once the bioreactor finishes, we would then proceed to collect and process the desired byproducts, terephthalic acid and ethylene glycol, which would then ideally be sold for use in cosmetics, the production of antifreeze, or even the formation of new PET or polyester. We have two main avenues for community engagement: social media and online events. For our social media page, we post regularly about what iGEM Toronto is doing and what we're learning. Using our social media platforms has given us the ability to share knowledge about what the textile waste issue is and how people can help. Building up our presence here has also helped us promote our events. We decided to host a synthetic biology panel with industry professionals to give students interested the opportunity to hear what it's like and how they can get involved. Similarly, in our discussion event, we invited campus groups to discuss the ethics of germline gene editing, as it presents a growing concern in the field of synthetic biology. This event then targets the general population, which hopes to teach them about the concerns surrounding gene editing.
In summary, we have designed a novel method of rational design that doesn't rely on flawed energy functions, a novel machine learning approach for generalizable de novo design of optimized proteins, and a set of enzyme variants along with their genetic architecture in a full bioreactor process with the potential to be used in large-scale industrial PET degradation. Our future directions include validation of our enzyme variants using our iterative procedure and its associated assays, testing the generalizability of our machine learning approach and its ability to optimize other diverse proteins, and to integrate our best variants into our MG1655 microbe and bioreactor, using our integrative pipeline to test their efficiency in PET depolymerization.